All right, so I'd like to welcome Dr. Madhan Tirumalai. He is a professor, a research professor at U of H on astrobiology, so this should be a very, very interesting talk. He's also a publisher of 18 papers, 11 as first author, and he also has something very in common with a lot of the rest of us. He's a volunteer at HMNS. Well, we are volunteers at HMNS, but he volunteers in paleontology. We volunteer in astronomy, so we're out at the George. So we have similar commonality there and interest there. We also, I think, have some club members who are also into fossil hunting, so I'm sure they find that quite fascinating. So without further ado, I'd like to introduce Dr. Madhan Tirumalai and his talk on astrobiology, a journey into the unknown.
Yeah, that's good. So, is it the right place to stand? Is it okay? Okay. Hello, everybody. Thanks for inviting me. And I see that it's a packed house. I'm very happy to see so many faces. So, okay. So, good. So he has already given me the introduction. I don't need to say anything further. It's been quite a journey. What I'm going to do today is not go deep into whatever I'm doing. It'll be like an overview, just spark anybody's interest. If we can then later on, this need not be the final presentation. I can magnify any of the themes that I will cover in subsequent presentations. If you would be happy to invite me again. Okay. So I'll begin with the first. This was supposed to be a surprise for everybody, but which one do I move? The wrong one? Okay. So you're all familiar with Leonard Lemma, right?
Yeah.
Yeah. Okay. Of course, everyone would be. So begin with the quotation. Is it possible to move this down? Because we cannot.
Otherwise, we have just a window.
Oh, okay. Anyway, I begin with this quote because in not just in astrobiology, in any branch of science, right? We are so closeted in our respective small niches, and we hardly have a time to interact or communicate our subject field to the general public. Around 2012, I received an invitation. An invitation was forwarded to me by my mentor, George Fox, to make a 3-minute presentation to a general public at NASA, at one of NASA's centers it's called the USRA building on the way to NASA. I thought, why should I be doing this? Why should I be doing something goofy like this? Because we're supposed to be doing serious science. But he said, there is nobody to represent our lab. So I did it. It's called the FameLab presentation. 3 minutes, it's an elevator talk. You speak about any topic in astrobiology or science to a general public without dumbing down the science and make it interesting. And it turned out, I realized I had a penchant for it. So one thing led to the other, and I have since then been making several astrobiology outreach presentations. And only 2 weeks ago, I had my first peer-reviewed publication on outreach. I would be happy to share with you guys that paper if you're all interested. But anyway, so this is one of my favorite quotes. The miracle is the more we have, the more we share, the more we have. So that's why I'm here to share what I think I know about astrobiology. Oh, we're going back. Okay. The journey into the unknown. How many of you have heard of the word astrobiology before? Okay, cool. Okay. So the NASA website talks about astrobiology as life here and beyond. It's a study of the origin, evolution, and distribution of life in the universe. Now that's a tall order because we have not completely understood the entire spectrum of biology and life on Earth. So why do they? Oh, what happened?
Oh, I have to reshare. I'm sorry. There's an issue with the share.
Oh, okay. Well, I've now become a star now. Thank you. I'm in the limelight. Okay, so astrobiology is defined as the study of the origin, evolution, distribution of life in the universe by NASA. That's a very simplistic definition. How do I move this? Okay, so it all began with with this fragment of a Martian meteorite which was discovered in Antarctica. It is called Allen Hills 84001. And when they discovered this, they presumed and assumed that some of the microscopic structures that they saw in this piece of meteorite could have only been possible. They made an assumption that this is possibly a result of microbial or some kind of a biological activity. And so, as it always happens in science, there are a lot of debate and discussions about this. And subsequently, it was concluded, or the consensus arrived by the wider scientific community, that this hypothesis could not be possible because all the unusual features that were seen in the meteorite piece could be explained without requiring life to be present. It could be a result of geological processes, geochemical processes, just plain chemistry. However, it's not all bad, because this particular series of discussions around this meteorite was kind of a seminal event which resulted in the emergence of the field called astrobiology. Okay, so the discipline of astrobiology had its nascent birth in the mid-1990s. And 1998, the Astrobiology Institute was founded by NASA, and eventually it has now evolved into what we call as the Research Coordination Network. Do not ask me why they keep changing the names, but the central theme is astrobiology.
Question?
Yeah.
What do they call it again, the new name?
It's called the Research Coordination Network. Yeah. So what does the field of astrobiology deal with? It's like a large umbrella under which there are multiple disciplines. Primary being the origins of life. So in the origins of life, one could look at the genomics, phylogenetic relationships between different forms of life. What is the relationship between plants and the bacteria or microorganisms associated with plants? What is their relationship with us? And what is the relationship between the bugs that are associated with chicken, the chicken farm, and us? And this has a lot of these kind of studies that are done in astrobiology have a lot of implications for something as diverse as how antibiotic resistance spreads between microorganisms, between different forms of life. But I will not go into that. That will be for another day, another talk. Okay. So that's one thing. Second thing is it's to do with life in extreme environments. How many of you are familiar with biology in extreme environments on Earth? Okay, good. Two. So you might be familiar with some of the things that I'm going to speak today. So bear with me. And then laboratory prebiotic experiments. What kickstarted life or what kickstarted biology? We do not know. There is no way we can go back in time and take snapshots of how things happen. However, we can look at molecular evidence as it is available now, not macroscopic evidence, but microscopic evidence. Look at the molecules, molecular evolution, and then go back in time. And then it's like, imagine if you're climbing down a ladder in time. You go to the last rung, you do not know what to do after that because it's just dark abyss. What you do, you then go back, go further down. What I describe now is top down and then bottom up. Bottom up, what they do, they create several models and chemical systems, and then let the systems do the talking, and then look at the data, and then come up with assumptions, and then see what kind of bottlenecks could have driven those pathways to the last rung of the ladder that I spoke about earlier. Okay, so for that we do something called laboratory prebiotic experiments, and then there are also computational work involved in which organic simulations are done, and then observation of the same. And then astrobiology also deals with planetary systems, other planetary bodies, and then the entire gamut of science dealing with planetary systems. And there's a special institute called the Search for Extraterrestrial Intelligence Institute, SETI Institute, which is in California. Please go ahead and take a look at their website. There's some very, very fascinating, interesting stuff. So what they do is they look for signatures, astrobiological signatures from other planetary bodies, which might be evidence for biology as you know it. And last but not the least, astrobiology field also, because NASA is funded by the public, taxpayers' money, there's an important component of outreach, which is what I do. I started this as a hobby, like fun. It's now become a serious thing. So all these fields, all these diverse fields are covered by astrobiology under the large umbrella of astrobiology. Okay, but I'm going to talk about, let me go one step back. So much of my talk today will be to do with life in extreme environments. So extremophiles, my PhD back in India was on a group of extremophiles called halophiles. How many of you have heard of halophiles? Good. So we have a young gentleman here. So yeah, thank you for being here. So halophiles are microorganisms that thrive high salt conditions. Okay, they love salt, the kind of salt concentration that if you had to consume, you're going to puke immediately. Okay, that's the kind of bugs I work with in my PhD. So since then, most of my work has been related in one way or the other with either extremophiles or astrobiology or origins of life. So let's see what these extremophiles are all about. Okay, this is something most of you would know about. It's called the tardigrade. The reason I put up this slide is make sure that I get away with this. So I get rid of this so that I can focus on the other things that people may not know about. So what are tardigrades? These are not bacteria. These are microorganisms. These are very close relatives of arthropods or, for example, insects and crustaceans. They are very, very versatile in where they live. They live in damp moss. They could live on plants, in sand, in fresh water. And they are known as water bears or moss piglets because they are less than 0.02 inches in length. Okay. Some of them may find them cute. They are indeed cute and they are harmless mostly, but they are extremely versatile. They can survive, for example, for a few minutes at minus 272 degrees Celsius. We don't even store our bacteria at this temperature. We store minus 70. Okay. Or they could survive as high as 150 degrees Celsius. That is 50 degrees more than the boiling point of water. Okay. And they could survive for decades at minus 20 degrees Celsius for, it's a typo. They could survive for decades at minus 20 degrees Celsius. For example, your freezer and your refrigerator. Okay. And they can be revived after that. Okay. They can also survive pressures from 0 atmosphere in space up to 1,200 atmosphere at the bottom of the ocean. And also the most extreme radiation levels from 5,000 to 6,200 grays. I'll be talking towards the end of my talk about radiation resistant bugs. But this is a classic example of an extremophile that can survive multiple conditions. So we call them polyextremophiles. They can survive multiple extreme conditions.
Now, when you say extremophile, you make it sound like it likes extremes.
Yes.
But if you've shown it can tolerate extremes.
That's a great question. Well, yeah, this word is often used in a much more broader sense. But in the strictest sense, extremophiles are those organisms that they cannot survive without extremophile conditions. Yeah. But thanks for pointing that out.
The tardigrades, they don't normally live in those conditions, right?
Yes, extremotolerant, you can call them. Yeah.
Okay.
Yeah, but that's a very, very important point because these are the kind of questions we get from students. And I'm glad that somebody asked this question. Okay, so what is an extreme environment? It's an environment that threatens access to basic requirements, the integrity of biomolecules, has limited resources for basic needs, and physiologically stressful conditions. I'm assuming that most of you have lived much of your life in Houston, right? Hot and humid, 4 months of the year. June, July, you can make an omelet on these roads, okay? The tar sometimes almost melts, right? You take somebody who has lived his or her entire life in Houston, put that person in a place like Michigan, near the lakes. I was in Michigan last week, so I know how cold that can be. So for this person who is from Houston, it can be extreme in Michigan. Take a person from Alaska who's lived his or her entire life near Denali National Park and put that person in the equatorial region, like the tropical regions, like, for example, in India or the Sahara. For them, that is extreme. Why is it extreme? Because their body is already used to a certain set of conditions and suddenly it becomes extreme. Okay? That is extreme as we define it. But as you will see subsequently in my further slides, how extreme is a word that we coin for our convenience for bacteria or bugs. Extreme is not what we think as extreme. Okay? That's the basic explanation for extreme conditions. Okay. sorry. Here is an example for, somebody asked a question?
No.
Okay. So some examples of daily use things that we use. If you take the egg yolk, you can either subject it to thermal denaturation, or you can subject it to mechanical denaturation. In either of these cases, what happens is when you subject them to this kind of extreme conditions, it results in the denaturation of the proteins, which is what you consume for your breakfast as an omelette, or eggs that are beaten or scrambled, right? So what you're doing, you're subjecting the biomolecules in these food material to extreme temperature or extreme physical agitation that disrupts their structure. Then it is consumable, then becomes edible for you. Well, it will be edible even otherwise, but it becomes, because we have something called a taste. We have acquired taste, so we do this. We make these things undergo these kind of denaturation by subjecting them to these extreme conditions. Okay, here is a chart that shows a different kind of spectrum, pH. Okay, at the lower end of this pH it's acidic, and then you have the neutral pH at 7, which is the water that we drink. These days it's a fad to drink water which has higher pH, alkaline pH. People make a claim that it is good for your gut bacteria. I'm not entirely convinced, because peer-reviewed research is not that convincing. But it's a free world. People can assume things and do whatever they want to. However, the point is the hydrochloric acid, okay, which is used in laboratories and many industrial processes, it has a pH of 1, or it could be lesser than that. Lemon can have a pH of 2, and then vinegar can have a pH of 3. Vinegar is nothing but dilute acetic acid, which you use as a dressing. Tomatoes can have a pH of 4, bananas 5, and then water 7. And then on the other end of the pH, as the pH increases, it becomes alkaline. Okay, your baking soda, all the way up to the kind of things we use for cleaning ovens. Okay. Biomolecules can get disrupted either the lower end of the pH or higher end of the pH. So for biology as we knew it 40-50 years ago, life was not thought to have been existing below 6 or above 9 pH, but it's all changed now. Okay, so how did we come about with the word? This will answer the gentleman's question in the back. The word extremophile is a combination of extremus, meaning the superlative of exter, meaning on the outside. Philos means love. When they combine both, the word extremophile was coined by Bob McElroy from NASA Ames. That's how this word was coined. Okay, so what are the extreme environments on our own planet Earth? Okay, where are they? What do they look like? Oh, what happened? It's not moving. Oh, okay, it's moving. Okay, these are the various extremes of extreme environments on Earth. One is extreme of pH, as I explained to you in the slide. It could be low pH acidic conditions or high pH alkaline conditions. It could be extreme salinity, as I explained to you before. My PhD work was on extreme salinity loving bacteria or bugs. It could be extremes of pressure at the bottom of the ocean. It could be extreme radiation. It could be extremes of temperature, low temperature, or known as psychrophilic, or a high temperature. Or it could be extreme dryness, we call it aridity. It could be any of these conditions, or it could be a combination of some of these conditions. That could be defined as an extreme environment. Okay, we look at some of the environments that have a combination of low pH, acidic, a high temperature, thermophilic. Okay, you're all familiar with this place. It's called the Yellowstone National Park. Okay, if this gentleman has never been to the Yellowstone National Park, you should ask your school teachers or your family to take you to the Yellowstone National Park. It is truly an adventure that you'll never forget. Okay, so the Yellowstone National Park is actually a caldera. Okay, it's on a caldera, which comes from the Spanish word caldera and Latin calderia, it means a cooking pot. It's an English term for a cauldron. Okay, these are depressions that are found in all volcanic environments on Earth and in the solar system formed during eruptions. So, what is a caldera? So here is a kind of a pictorial depiction of the same. So the Yellowstone caldera is like this. It's basically like a piece of land on top of a boiling mass, and there are fissures on the earth through which steam and other material from the bowels of the earth come out at high pressure, and this is predicted that at any time this might erupt. I hope it doesn't, but you never know, right? So this is Yellowstone. Because this is characterized by these fissures through which very hot plumes of air and water come out, and when they come out, because they interact with the cooler temperatures, they mineralize, they form all kinds of strange structures. Okay, this is a picture I took of the date is wrong, the time is wrong, because there's no way I'm going to be inside the Yellowstone National Park at 3:31 a.m. This is more like 11 a.m. I didn't set my camera right. So this is, you can see here, one of those water bodies there. You can see this yellow structure. These are all, this is sulfur compounds flocculating, okay? And all these things are rich in microorganisms, okay? This is one of those geysers there in Yellowstone National Park, and surrounding this, you'll have many layers of different communities of microorganisms. This is another picture that I took, and you see these mats here. These are actually microbial biofilms that have been formed over millions of years, and they have a huge role in the biogeochemical cycle of the Yellowstone National Park. So it is not microorganisms, combination with natural chemistry, they play an important role in formation of those structures in Yellowstone. Okay, you can see here, hot water will scald, danger. They put these signs throughout. All these structures, right, terrace-like structures, they were formed by a combination of chemistry and microbiology. And the water is extremely hot and acidic too, okay? Apparently, a few years ago, somebody fell into this and lost their life. Yeah. Yeah. This is just another view of another part of the park. Okay, you see these structures here, right? So these structures, as I explained to you in the caldera picture, these structures have been formed over millions and millions of years of geological evolution. When the hot material comes out, it cools down and then it condenses to form these structures, along with the microbial activity. Okay, this is a closer view of one of these structures. Another view. Oh, okay. Yeah, it would be really nice if we can move that to the bottom, but it's this whole thing. I think this is the way to do it, right?
This thing moves.
Yeah, it moves. You just have to select it. Yeah. Awesome. Thank you. You made my day. Yeah. So when the hot water containing sulfurous compounds, it comes out, it cools and causes precipitation. This is all making these little ledges. So what we are seeing here did not happen yesterday or a few years ago. Millions and millions of years of evolution, years of geological evolution. Okay, and in these structures, as I said before, thermophiles inhabit and may assist in creating these unique structures, besides geochemistry alone. So why are these structures important? They are important because when we have flyby missions of other planetary bodies, we look for structures like this. And if there are structures like this, the first question that's unusual, we try to compare them with structures in Yellowstone National Park or the Atacama Desert, about which I'll be talking to, and then figure out, could these structures have been formed by alien bugs? Don't ask me if they exist. We do not know if they exist. As of now, it is fingers across, but it is the assumption, astrobiology is also a science of hope, it is the assumption that perhaps there might be life. And if there is biology elsewhere, similar to what we have on Earth, perhaps they'll have things like this. Oh, okay, what happened? Oh, okay. So here is an example of some of the organisms that actually live in biology that lives in lower pH and their pH limit. Fish can survive up to 4 acidic. 4, if you recall the earlier slide, 4 is the pH of what? Lemon, right? Insects can survive up to 2. That's 2 is extremely acidic. Some plants like mosses, vascular plants can survive up to 3 or 2.5. Eukaryote or higher microbes can survive up to 1 to 2. Fungi 1 to 2. Prokaryotes is actually an incorrect word. Prokaryotes refers to microorganisms like bacteria. Okay, they can survive up to 0.8. Here are some examples: Thiobacillus sulfolobus. These are not bacteria; they belong to a third kingdom called Archaea, but they are all microorganisms. Okay, they can survive or thrive at pH as low as 0.8. Okay, this is the kind of a pH that you don't want to go near. It's the kind of a pH you find in some of the extreme conditions in Yellowstone National Park. And those places are thriving in microorganisms. So here's a real cool slide. You see here, every layer of color, it's almost like a painting, right? But every layer here, every color is formed by a distinct microbial community that inhabits, because when water comes out, it is not almost always in the same temperature. So each color represents a microorganism that has adapted to that particular temperature and gives this absolutely beautiful artist palette-like thing. This again is from Yellowstone National Park. Okay, some more examples of these microorganisms. All of them have this word sulfur attached to their names, except this one. This is, again, it says the word acid because they're all is acidic. Sulfur means they either use or they use one of the sulfur compounds as a source of energy. Okay, that's the reason why in one of those pictures you saw those sulfur flocculating in the pools. Okay, and they all have a pH optima of anywhere between 2 to 2.5 or even 3. And the temperature, only this one has a temperature optima of 75 degrees Celsius. The rest of them, in general, if you take a bacterium like E. coli, which you may have heard of, which lives in our gut, and which is also everywhere in the environment. If I were to do a microbiome analysis of this room, I'll probably find E. coli, which has come from us, from our skin. Then there'll also be another organism called Bacillus. Their maximum thriving temperature is 37 degrees Celsius. So for them, this will be extreme. But for these organisms, this is the temperature that they want. Okay, now we'll go to a place called, a place in Japan. Japan is formed by a series of volcanic islands, right? So it's a place called Mount Yoti. And you see this? He's probably an astrobiologist or a geologist or a volcanologist. All this yellow that you see here, it's all from the sulfur. Okay, and 2 organisms have been found here in this extreme environment. They are named Picrophilus oshimi and Picrophilus torridus. These are extreme thermophiles. Oh, what happened? Oh. Okay, and then we move on to a place called Redding in California. How many of you are familiar with what is going on in Redding, California? Okay, so Redding used to be a place where they had acid mines. Okay, it's a place famous for mining. So what happens is you have this mine ore material in the bowels of the earth. You suddenly expose them to oxygen by digging them, and microorganisms start adapting to those conditions, and then they actually start leaching them out and lower the pH. So these are extremophiles that are not only adapted to, they actually cause the acidification of these acid mine drains. Okay, and in California, apparently this is a huge problem because microorganisms contributing to the acidity of these drains is actually resulting in these water seeping into aquifers, which humans consume. And Therefore, they need the help of microbiologists and geologists who work hand in hand to figure out if they can come up with other microbial systems that can counter this. Okay. Here is a sample of what, okay, according to EPA documents, workers by mistake, they left this shovel standing in a green liquid flowing from one of the mine portals. Okay. Just overnight when they came, the shovel had been eaten away by the acidic drainage. And that acidic material was created by microorganisms that live there. Okay. This is just to show that the definition of what is extreme is limited by our own imagination, not by how life thrives. Okay. Here is an example of another organism that I'll be speaking about. You see this reddish thing. This is all not sulfur, but iron compounds. And there is one particular organism that is which is very dominant here. Okay. It is called an acidophilic ferroplasma species. Okay. That lives in that, you see here, that lives in these conditions. And you see this, most people will mistake this for, a zoomed in version of peacock feathers from a bird, but they are not. These are actually biofilms. So for those who are not familiar with the word biofilm, a biofilm is nothing but a large community of microorganisms that exude polysaccharide material. So it's like a slimy material. They do it because when they grow in a community, it is better to have access to food that is equally distributed to everybody so that they can reproduce more. Okay, that's a very crude explanation for a biofilm without getting too technical. So this is an example of a biofilm made by the acidophilic ferroplasma species. And what is cool about this, it can grow at a pH between 0 and 0.5. Now, this is crazy. This is as acidic as it can get. Okay, and 40 degrees Celsius. And these are found in streamers attached to pyrite surfaces at a sulfide ore body. Again, sulfide ore mine body. Okay. So what has happened is, before the mine was made, right, this organism was probably in some small niches where it was acidic. But as the mine was exposed to the air, and this particular organism, it started dominating the others, and it moved everything out, and it became the dominant species in that ecosystem, in that small ecosystem.
How long are the green streamers?
What was the question?
Are the microscopic? Is that microscopic?
Well, no, sometimes they will not be microscopic. Classic example, when you get up in the morning, look at your tongue, the white lining, white thing that you see, which you use a tongue cleaner to remove. That is an example for a biofilm.
Let me make a comment. 40 Celsius is equal to 104 Fahrenheit. That's about as hot as a jacuzzi.
Well, actually, 40 degrees Celsius is not that extreme, given the examples I gave, right? So sometimes they can survive up to 100, 105. And I'll give you more extreme examples as we go forward. Any more questions?
I do know that 100 Celsius is 212 Fahrenheit. That's when water boils.
Yeah, that's the boiling point, yes.
So just follow up on the questions that was asked, what scale of objects are we looking at right here? So
This should be, well, haven't put the scale. This should be visible to the naked eye. Yeah. Okay, this is an iron oxidizing bacterium. It's a dominant organism in this environment. It is dominant, and it has adapted so nicely that it constitutes 85% of the microbial community there. Okay, now let's take a dive into the oceans. Okay, and talk about something that most of you might be familiar with. They call the hydrothermal vents. There are some very unique geological features. So what does the ocean floor comprise of? So those who are in school might find this really exciting, because next time when you go to the ocean, you may want to know how these things are formed and how the ocean structure has been formed and continues to evolve. Okay, so this is a classic depiction. You have what is known as the intertidal zone, which is near the shore. You have the reefs, the coral reefs. Most of it is shallow seas. For example, on the west coast of India, we have around 200 kilometers, which is what, 160 miles of this shallow seas. And then this is called the continental shelf, and then you have the continental slope. Okay, at the bottom of the continental slope, you see where you have the seabed. Remember what I told you about the caldera? Take the same caldera, put it at the bottom of the ocean. You get the hydrothermal vents. Hydrothermal vents are the equivalent of what you find in Yellowstone National Park, but this time under the bottom of the ocean. Okay. Any questions? Okay. What are they characterized by? They are characterized by low pH, highly acidic, drastic temperature fluctuation within a small area, and very high pressure, and high concentration of metals as well. So each of this is an extreme condition. Okay. For example, high concentration of metals. I was in Michigan last week in Flint. What is Flint known for? Lead, right? Lead contamination. So lead contamination or arsenic contamination, cadmium, these are not good. And this ecosystem is rich in all kinds of metals. Very high pressure, low pH, and drastic temperature fluctuation. Oh, what happened? Why is it not moving? Yeah. So underwater volcanoes on the seafloor produce hot springs, equivalent of what you find in Yellowstone, and they are known as hydrothermal vents. Okay. So where are these found? So this is a picture that shows the location of the hydrothermal vents, mostly along these tectonic plate borders. They are found all over the world in these locations. Okay. And again, a few more words on the conditions there. It's an absolute lack of sunlight, so they cannot utilize sunlight as a source of energy. So the chemical energy is utilized from sulfur compounds to fix carbon dioxide, and the waters can be superheated up to from 60 to 150 degrees Celsius. Right? We spoke about the temperature in jacuzzi. This one is at least 60 up to 150 degrees Celsius or even more. Okay, extreme pressure. The more the depth, the higher is the pressure, and highly acidic. The vent plume, when it comes out at a very high pressure, it has a pH of around 2.8. So we'll go to a place called Tonga Islands, which is which is in the Pacific near Australia. And these are examples of some white smokers. So they're called either white smokers or black smokers based on the composition of what is coming out. And I think this is a video. Let me see if I can play this video. Yes. Can you increase the volume?
Well, that was our assumption.
Yeah, so this is an example of how, what kind of an extreme pressure, the things come out. You can see the things coming out at a very high, at a rapid pace. So it is under these conditions that thermal vents are formed. Okay, let's move then to a place of the Pacific Northwest. It's called the Yuande-Fuca Ridge, which is a geological wonder for geologists. And this is home to a huge underwater, almost like a town of hydrothermal vents. And what is found there? You have this smoker, black smoker, hydrothermal vent called Sully, probably named after a pet dog or a cat by the person who discovered it, Sully. And it emits jets of particle-laden fluids. And it's at, move that around. Yeah. It's around approximately 2,200 meters. So keep an eye on the depth now. as I move on with the slides, the depth is going to increase. Okay, it's on the Fuca Ridge. What you see here, right? All these things, these are all tube worms that are found thriving here, and these tube worms have microorganisms associated with them, living in harmony in a symbiotic relationship. You know, I give you something, you give me something. The give and take, a symbiotic relationship. Okay, the temperatures, look at this, 300 to 400 degrees Celsius. Okay, you don't want to go near there, right? 300 to 400 degrees Celsius, and the pH value is 2 to 5. Okay, and because the hot acidic fluids transport very high concentrations of metals, like iron, copper, and zinc. I have left out a few others like cadmium is also found. When this kind of fluid mixes with seawater, which is cold, the result is a black smoke of all these metal sulfides getting precipitated out. Okay, now in Yuande Fuca Ridge, there is this one organism, microorganism called Geobacterus. Okay, it belongs to Geobacterus class. It is found in a deep sea hydrothermal vent called Finn, around 200 miles off Puget Sound, and it is this particular place where it is found is around a mile and a half deep, and the temperature at which it is found ranges between 85 to 120 degrees Celsius. Okay, this is the organism. And what you see here, these are all branches of what is known as flagella, with which microorganisms move around. Okay, isn't that cool? Okay.
Bacteria, said geobacteria.
Yes, they are bacteria. Yes.
Can you say anything, doesn't have to be detailed, Yellowstone, Wyoming, and hydrothermal vents, one of the things they have in common besides temperature is this high acid. Okay, can you say anything about the chemistry that's causing that?
Part of the reason the chemistry is that way is because of the microorganisms themselves.
Oh, they're causing.
They're causing that. Some of them are causing, some of them are adapted to them.
Okay, very good, thank you.
So let us go to high pH alkaline conditions. Now, in the case of high pH, so I'll explain it to you now itself. It is because of the geochemistry that causes the increase in alkalinity. It is not the microorganisms. Okay. We will look at hypersaline, high saline as well as alkaline conditions. How many of you have ever been to this place, Kilimanjaro? Right. Next time you pay a visit to Kilimanjaro, not far from here, most people will think of going on a safari to look at the lion prides. But not far from here is a place called Natron, which is at the border of Kenya and Tanzania. This is a lake. Do you see this? It's a shadow of a plane. Most likely the plane from which this picture was taken. All this red color that you see is formed by both microorganisms as well as slight eukaryotic organisms, more complex organisms. And the red color is imparted by the pigments produced by these microorganisms. Okay. And these places are characterized by, this particular place is characterized by hypersalinity as well as higher pH of 9 to 10.5, almost as alkaline as a pure ammonia. Okay. This is the kind of alkaline pH in which you use your oven cleaners at. Okay. And these organisms have tolerance or even thriving at extreme high salt and alkalinity. And okay, here is another view. This is to give the contrast between the color on the lake and the color on the green shore side. Okay, it's called Lake Natron. And it has an unusual mineral content leached from the surrounding volcanoes, which is contributing to the alkalinity. Okay, we'll move to a place of California, which is called Owens Lake. You see this red pigmented coloring, that is again from halophiles. If you ever fly into the Salt Lake City or Great Salt Lake, you'll find the edges of the Great Salt Lake will have this color. It's from extreme halophiles. It's these kind of organisms that I work with.
The place in Australia that has kind of a pink, bright.
Yes, it's called Lake Tyrrell, if I'm not mistaken. Yeah, I may have a slide, or I might have. Yes, I might have removed it. I hope I have the slide. Yeah. Okay. So what are extremophiles? Extreme halophiles. Extreme halophiles, they require large amounts of sodium chloride for growth. I'll give a small daily life example. You take a glass of water, right, from your tap, put your finger in for 10 minutes. Maybe your finger your skin might shrink. But if you add, let us say 5-6 teaspoons of salt, okay, put your finger in and then see what happens. All the cells in your finger will lose their biomolecular structure that are inside them, and they'll undergo a condition called osmolysis, where the cells will lose the water to equalize the salt concentration. So it's an extreme condition. These organisms require large amounts of sodium chloride. How do they do it? They balance the high salt concentration by accumulating high levels of compatible solutes, such as potassium chloride, to balance out the sodium chloride outside. They either use potassium chloride, they use other compounds like glycine betaine. I don't want to go deep into those. I use this here so that you will all be familiar with it. There are other compounds they use to balance the molar concentration. Okay, here is an example of one of the pigments that causes that results in that color. It's called halorhodopsin. Okay, believe it or not, halorhodopsin is related to a protein in our retina. Okay, if you were to draw a phylogenetic tree of the protein sequence from our eye in humans and take this halorhodopsin, you will find some similarities. They all had a common ancestor. So did we get our opsins from bacteria or archaea? We do not know. That's a question future PhD candidates like that kid might answer. Okay, halorhodopsins are light-mediated proton pumps that help extreme halophiles make energy. And now I'm going to take a jump to talk about other worlds. Okay, what about Mars-like conditions on Earth? Okay, so here is a picture that shows a comparison of Earth with Mars. Since you're all astronomers, come to actual the relationship between what I said so far with astrobiology. So why is Mars so cool and so important? Mars has these following features. It lacks a global magnetic field, and it has a very thin atmosphere. The combination of these two, the very thin atmosphere permits significant amount of ionizing radiation to reach the Martian surface. That's one of the biggest challenges that will be for either NASA or Elon Musk or anybody else who wants to live there, right? How much, am I good on time? How much time do I have? Oh, okay. Yeah, I should be done. Okay. Mars is also is important because they have discovered salty brines. Salty brines are actually subsurface reservoirs of high salt conditions, very similar to hypersaline reservoirs, hypersaline ecosystems on Earth, and reservoirs of liquid salt on Mars. Trace amounts of methane, which varies with seasons. Okay, so the question is, where is this methane coming from? Is it a chemical process or is it a microbial process? So the hypothesis is that microbiologists are excited about is maybe it has a microbial origin, but I don't think that's the case, at least so far. And Martian soil contains perchlorate. Okay, these are salts derived from perchloric acid, and perchlorate is also found in places like Nevada, Mojave Desert. Okay, it is the most Earth-like planet, second closest planet. It has Earth-like seasons, as much land surface as the Earth, abundant water, salts, volatiles, and metals, and carbon. Okay, this is the city of Austin for scale. Okay, this is a picture. I think a gentleman from the Bureau of Economic Geology. He shared me some of these slides in one of my astrobiology presentations. Okay, just to show you all how much ice is present on Mars. Okay, these are Mars-like places on Earth. There is something called these are analogs, as we call them. Wet Mars in Canadian Arctic, frozen Mars in Greenland, wet Mars in a place called Rio Tinto, dry Mars in the Atacama, wet Mars in a place called Pilbara in Australia, and then Calgary, and then dry Mars in the and frozen Mars in the Antarctic and landmass. Okay, it's another slide. This is actually from a recent paper that I was talking to you about. Canadian Arctic, Yellowstone National Park, about which we already spoke about. Rio Tinto, Atacama Desert, I'll be speaking about it shortly, in Australia, and then Antarctica.
What country is Rio Tinto in?
Spain. So if you, I don't have the pictures here because otherwise my talk will go to 3 hours. Rio Tinto, if you see the pictures, you can Google it. The rivers are like yellow red. Yeah. Okay. So let's go to Antarctica. What do we find in Antarctica? It's thought to be, apart from people going there, most people thought it is like uninhabitable for microorganisms. Here is an edge of I just put these pictures because I thought they were really cool. Okay, here is an example of a deep lake in Antarctica, 50 meters below sea level and never freezes, and there are microorganisms found in this subsurface. Okay. Yes. Oh, the water never freezes? No idea. What a great question.
It's the salt, right?
It is because of the salt, or it is because it doesn't freeze, there is salt. We do not know, right?
In Chicago, where I come from, in the middle of winter, when you have ice on the ground, you put salt on it, it melts all the ice.
Oh,
This is deep below the ice, so this is, I think, near the actual ground and out between the ice and.
yeah, maybe it is because, yeah,
It's a subterranean lake, about subterranean exposed to the surface.
yeah, that could also be the reason, along with the combination of salt, because
They drilled down the deep lake.
this, I don't think this is hypersaline per se. It's just, I think it is a deep, okay, it's a salt lake, yeah.
There
Yeah.
are lakes that are, they're covered with a mile of ice, and they're warm, just by the earth warm coming up, I think.
That probably explains, yeah. Okay, so these are examples of papers and some of the things that they found. They found prokaryotic. Prokaryotic refers to microbial diversity. These are metabolically active communities in brines, high salt environments, at a depth of 0.9 to 2.5 meters. Temperatures anywhere between 31 degrees to 0.9 degrees Celsius. So these are not exactly extremophiles, they're extremotolerant. Okay, and this is another example of microbial life at minus 13 degrees Celsius in the brine of an icy Antarctic lake. And if you go further up north, there are microbes found in sub-zero hypersaline brines in a formation known as cryopegs. I don't even know how to pronounce this place.
Barrow. I'm joking, it used to be Barrow.
Oh, okay. Ukiawik. I don't know. Maybe I got that right in Alaska.
Name what the locals call it. And I don't know what that's.
Yeah. So you have sub-zero brines at minus 6 degrees Celsius, 11.5 to 14% salinity. And these are unfrozen sediments. It is known as permafrost, which is what you find on Mars too. And the dense bacterial communities can be abundant up to 108 cells per ml. And then let's go to the Atacama Desert, another analog. So it is an analog, for example, of dry Mars. Okay, and where is Atacama Desert? It is found in Chile, South America, and it is one of the highest deserts, probably the highest desert on Earth. And it is a desert because of the mountainous formation that blocks the rain-bearing clouds from coming there. You can see a comparison of images taken by the NASA Mars rover, and compare it with the Atacama Desert picture, you see some uncanny similarities, right? Besides the conditions, even look-wise, it's like Mars. Okay. Is there a way we can move this around? Yeah. See, this is just to show the level of precipitation that's there, okay? For over a 4-year study, a single rainfall of 2.3 millimeters took place, okay? Compare that with the rainfall we had on Thursday. Pretty significant, right? It's one of the driest and oldest deserts on Earth. Hyper arid for the last 90 million years, okay? If you were to compare this with Harvey, which most of you might be familiar with, okay? Harvey dumped 52 inches of rain over the Houston neighborhood in August 2018, okay? For the same thing to happen in Atacama, it will take 2,250 years if it rained at all. Okay, that's the comparison to show how dry it is. What's happening? Okay, characterized by low oxygen, high UV radiation because it's a high desert. Desiccation means very dry, and it often has winds and very cold temperatures. The question is, does the Atacama harbor life despite its hyper-arid conditions? You may want to ignore this figure because this is from another presentation for high school students where I told them a story taking an example from science fiction. I forgot to remove that, so forgive me for that. Yeah, there are fossilized stromatolites. So what are stromatolites? Stromatolites they're often called living fossils. Some call them living fossils. These are nothing but large levels of biofilm growth deposited over minerals, and then mineralization takes place, and then biofilm, again, they get fossilized over millions of years of evolution, okay? These are structures created by sediment trapping microbial mats. Okay, here is an example of some of the stromatolites in the Atacama Desert. And you see these in this particular. I don't think so.
Okay.
That's why they're called fossilized.
Okay.
And you see here, and this is under the rock. These are called, these are referred as endolithic sometimes, okay, or endolithic organisms, which can, in spite of the lack of water, they are thought to grab moisture from the fog that often comes near the mountain. They are still trying to figure out how they actually do that, okay? But the point is, there is microbial life in the Atacama, okay? Fog advancing towards the hill seem to be the main, if not the sole source of these microorganisms, source of water for the hyperlithic communities. Okay, and then from the Atacama Desert, let's move on to a place closer home. It's a place called Death Valley National Park. How many of you have been to the Death Valley? Cool, okay. It really looks like Mars, okay. It is the hottest and driest place in North America, averages about 1.58 inches of rainfall annually, okay. And it is this dry because of these three mountain ranges, Sierra Nevada, Argus, and Panamint, which make sure that rain-bearing clouds never come here. And it's a very strange geological formation, one of the lowest points in North America. And it's super dry, okay? Here's a picture that I took. Yeah, because of the rainfall, right? Yeah, yeah. So if you ever make a trip to the Death Valley, if you plan to, please go there and spend at least a week there. Not now. It might actually this might be the right time. Definitely not during June, July. So because of the rain shadow effect, you have these dry conditions, and also this is a picture of the windblown sediments on Mars. You can see a lot of similarities if you go to a place called, I think, Zabriskie Point, where if you look through your telescope or your camera, you can see that the surface remains one of some of the pictures taken from the Mars surface. And here is an example of microorganisms found in a place called Badwater Basin, which is the lowest elevation in North America, Death Valley. I'll play a video from this. So these are bacteria there are magnetotactic. It means they love magnets. These are not extremophiles per se, but they are found in the Death Valley, so they have to be, you can call them extremophiles. One of the unusual features is they gravitate towards magnets. That's why they're called magnetotactic bacteria. Okay, these are novel magnetite producing magnetotactic bacteria, belong to a certain class called gamma proteobacteria. Here is a picture of the same. So these are,
Yeah, I mean, they produce that so they know which way is up and down.
yes. And what is,
They migrate up and down, the sun will come bright, but they use the magnetic field to know what's up and down.
yep. So here is a picture showing the picture that I showed you. It's a greigite containing magnetotactic bacterium. So greigite is actually is made by these kind of bacteria. Okay, from that it's an iron sulfide mineral formed by these kind of bacteria and sulfate reducing bacteria. Okay, I want to play this video. Yeah.
Do you need to wrap up soon?
Yeah, sure. You can see the bacteria changing direction with the change in the direction of the magnetic field. We'll go to the next one. Yeah, play this also. Yeah. Yeah, you can see they're moving this way. When you change the direction, they go the other way.
The living compass.
You could say that, yeah. I didn't even think of that. Okay, we can, yeah. So Furnace Creek, it's an elevation of 190 feet. I just put a few more slides so that it's more interesting for those who might get bored. So it was found so cool that Star Wars is actually shot here in the Death Valley. That's RTD2 wheeling its way. And it resembles Mars so much that a mock-up of NASA's Curiosity Mars rover was actually tested here on Death Valley. Okay. The last environmental condition similar to human-made environmental conditions similar to Mars is a spacecraft clean room facility where spacecraft is assembled. Okay. Why is it called so? Why we think it is so? More than 30 years of extreme radiation, peroxide treatment, and desiccation, high dryness, has created Mars-like conditions here. And why do they do this? Because of a protection policy called planetary protection, and according to which, how can we protect other worlds from microorganisms found on Earth? If you're going on a flyby mission that falls onto Mars, or if he man mission, what if he transports some of our bugs from here? And how can we prevent microbes from other planetary bodies coming back here? Okay, coming back here, and those going from here there. Okay, so forward contamination and backward contamination. In order to prevent forward contamination, they have some stringent procedures. Try to finish real quick. Yeah. They often subject these facilities with UV, peroxide, and gamma radiation. Here are some examples. You can see that most of the scientists there are dressed up not like ordinary engineers, but more like surgeons and astronauts themselves. So the nutrient-poor environment, controlled moisture, and other conditions has created a special habitat for multi-resistant bacteria. Okay, this is from 1957. 1967, they actually had papers talking about microbial burden on these spacecraft before they were sent out. So the problem is, in spite of all these stringent conditions, we have actually, remember the acid mine drainage? We have created a special environment for evolving certain kind of bacteria. As a result of which, a number of novel microbial species that show high resistance to radiation, desiccation, peroxide, and gamma radiation have been isolated. And the question is, one big example is an endospore forming bacillus species with which I worked. I worked on the genome. It is found predominantly in the spacecraft, and I'll speak about it during our discussion. I'm running out of time now. So here is an example of some of these spores produced by this bacteria. They are super resistant to gamma radiation and UV radiation. Some more pictures of the same. Okay. So the question is, these unusual resistant properties of these contaminants, are they telling us that these facilities are breeding grounds for evolving these? And if that is so, could these ride piggyback on some of our spacecraft and contaminate other planets? That is one of the biggest concerns of planetary protection. Okay, so my final part of the talk, what other organisms do you know that are super resistant to high radiation levels? So in general, there is a radiation spectrum, just like the pH spectrum. The larger the wavelength, long wavelengths, it's not a huge issue, but the shorter the wavelength gets, the higher the frequency. You have ionizing radiation, which are cosmic rays, gamma rays, and then ultraviolet radiation, and then infrared, and then longer wavelengths, okay? So whether it is ionizing radiation or UV radiation, they are not good for your system. So DNA and proteins are the major targets of radiation. And when DNA gets damaged, right, it gets repaired. If they are in equilibrium, there is not a problem, okay? However, when the DNA damage rate outcompetes the DNA repair rate, then we have a problem, okay? So the vast majority of organisms on Earth are radiation sensitive, in the sense like you and me. They can be killed by doses less than 500. Gray is a unit. I wanted to keep an eye on this unit gray, okay? What other organisms? Last 3 slides. Now, what other organisms do we know that are very resistant to high radiation? Okay, take the example of this gentleman here. He cannot survive more than 5 grays of radiation, okay? However, an E. coli bacterium can survive up to 800 grays of radiation. So tell me, who is superior, us or the bacterium? And then you have an organism called Pyrococcus furiosus that can survive up to 2,500 grays of radiation. And then you have Deinococcus radiodurans, another microorganism that can survive up to 5,000 grays, 5 to 5,000. And it doesn't stop there. You have this organism called Crochiodiopsis, which can survive up to 4,000 grays. I should have changed the phone color. 4,000 grays of radiation. And then there are some examples of halophiles in which the resistance to desiccation and radiation are thought to be interlinked. Okay, and this has an implication for Mars hypersaline subsurface lakes. Okay, and then there is this organism found near Portugal that are resistant up to 11,000 grays of radiation. 5 grays can kill a human. 11,000 cannot kill this. Okay, and the last but not the least, the mother of them all is found in the Gaimas Mason. Okay, okay, 30,000 grays of ionizing radiation. I want you to think about this. 5 gray of radiation can kill a human. Even at 30,000 grays of radiation, this organism doesn't die. It means that at radiation levels in which the DNA, the genomic material should get obliterated, this organism has managed to evolve mechanisms to survive the same. Okay, so in summary, life has evolved to thrive in environments that are extreme only by limited human standards. The boiling acid of Yellowstone, miles beneath Earth's crust in pure salt crystals, and inside the rocks of the dry valleys of Antarctica. Okay, tomorrow this all has implications for our journeys to other planetary bodies, and this is not the end. As we endeavor to keep pushing our frontiers. And last few slides, we landed on the moon, so I put these slides. So anybody who wants to be an astronomer, an astrophysicist, astrobiologist, this is a great time because many countries are embarking on these missions. Okay, and that's it. Somewhere, something incredible is waiting to be known, Carl Sagan. So don't stop dreaming. Thank you.
That was really interesting. I actually have a question. Can you hear me?
Oh, yeah.
Okay. What do these extremophiles have with respect to their genetic makeup that enables them to exist in extreme environments? What I mean is, what protein structures do they share or have in common with other extremophiles that prevent their proteins being denatured? Do they share something in common?
Oh, okay. So let me see if I got the question right. The question was, do they have something special in their genetic material? Is that right?
Yes.
That's right, to enable them to withstand extreme conditions. Is it a common thing amongst extremophiles, or is each case different?
Yes. So, for example, the genomic content, our genomes are made of 4 bases: guanine, cytosine, adenine, and thymine, right? So
Yes.
these
Yeah.
organisms, they are far more GC rich than AT rich. That is
Right.
thought to be one of the adaptation mechanism. And also they have systems like chaperone proteins. So chaperone proteins are specialized protein molecules which forms complexes, and they kind of offer some kind of a protection shielding from all these conditions. They shield some of the major machinery in the cell. And
Right. And do all these extremophiles share the same thing?
yes, some of them share. Some of them are very specific to that particular environment. For example, halophiles, they will extreme halophiles, they will have their protein makeup will be highly acidic to because of the salt condition. Okay, and they have evolved these pigments which are very specific to the environment because they don't have access to nutrients. So they use these pigments to make energy from sunlight. So that is very, very unique to them.
Yeah. So they photosynthesize, but in a different manner.
Yeah.
So GC is 3 hydrogen bonds and AT is 2, right?
Yeah.
So it's a tighter, the DNA is more tightly held together.
Yes.
Okay.
So the more, in fact, it helps them with the rigidity of the structure. Even if you look at the RNA structure, the end of the end of a stem will have, if it has more stability, you'll always see a GC closing pair.
Sure.
Yeah, but to answer the question further, it is still an ongoing field. We know that there are chaperones. We know that the genetic makeup is different. Their constitution is different. We also know that most of these, they are archaea. Okay, they belong to the third kingdom of life called archaea, and they have a different cell wall membrane makeup than bacteria. Okay, they have almost like hydrocarbon-like material making up, whereas we have phospholipids. They have ether-linked lipids, similar to the hydrocarbons that is found in petrol.
I think I remember that Deinococcus radiodurans has 8 copies of its DNA. And so part of its secret is that it error corrects among 8 copies instead of just 1.
Yes, that is another thing. Their DNA repair mechanisms in general are far more efficient than any other the non-extremophilic organisms, particularly those living in high temperature, high radiation.
Like in human, break both strands, you're screwed. The Deinococcus radiodurans, it goes one of the extra copies uses that.
It's like a giant cluster computer where one thing fails, they use another. Yeah. I think she had.
Do they have the? Sorry, can I ask a question again? Do they have the ability to repair themselves if they do suffer damage?
Oh, I didn't quite understand the question.
I said, do they have the ability to repair themselves if they suffer damage?
So the primary repair mechanism is the DNA repair mechanism. Okay. For example, if the DNA is damaged, that's it. There's no point in even repairing anything else because nothing else is good. So that is a primary thing that they want to protect, right? The Thermococcus gamma tolerans, the example I gave towards the end, that is an organism that I would love to work with if I had the funding because it's an incredibly interesting organism. They are still trying to figure out how it can withstand this high level of radiation. And apart from chaperones, I think some metals could also be involved in protection mechanism. Though, yeah.
Thank you.
Yeah, you're welcome.
Okay, folks, we're running a little bit behind, but I want to thank Dr. Tirumalai for a very presentation.